Hello, everyone, and welcome to my presentation, where I'm going to present about the selectivity of protein interactions stimulated using terahertz signals. There is plenty of room at the bottom. This was the title of the lecture given by the famous physicist Richard Feynman in 1959. Feynman described his vision on the possibility of manipulating individual atoms and molecules to realize increasingly tinier yet powerful practical man-made devices in the future. In his talk, he noted that the engineering community should rethink the way in which these devices are conceived. In the last decade, researchers have investigated paradigms in order to initiate reliable communication between nanomachines, since traditional communication technologies are not suitable due to both the size as well as power requirements. The two most common intra-body communication techniques are molecular communication and terahertz nanocommunication. Molecular communication entails utilizing chemical signals as carriers of information, where it basically relies on a diffusion mechanism. Nonetheless, the emergence of nanoelectronics and nanophotonics have paved the way towards electromagnetic communication among nanodevices, where the terahertz band is a possible candidate. In today's presentation, I'm going to focus on terahertz nanocommunication. To give you a background about terahertz intra-body communication, it started in 2010 with the advent of the Internet of Nanothings. In fact, having a system where we have nanosensors, those nanosensors are able to autonomously transmit their sensing information, be controlled from a command center, or coordinate joint actions when needed. The potential of intra-body communication has been fortified with the characterization of tissues in the terahertz band. Then, in 2020, when we started our research, we raised the question, can we regulate protein interactions using terahertz waves? But two questions which are going to follow: why proteins and why proteins and terahertz? Well, as a start, proteins facilitate signal transduction processes. They are basically the front line of many signaling events. Proteins are going to also allow us to bridge the gap between electromagnetic communication and molecular communication. Why proteins and terahertz? Well, proteins exhibit vibrational spectral features in the terahertz regime, corresponding to functionally relevant modes. These modes are regarded as the possible dynamics, leading to conformational changes and biomolecular functions. So, a question that arises, can we basically trigger those modes? Let me walk you through our system model. In our system, we have a transmitter, which is basically a terahertz nano antenna. It's tuned to the vibrational frequency of the protein. Our channel is basically stochastic, composed of intra-body particles, and our receiver is going to be the protein or protein population, which is going to acquire a folded conformation upon being triggered by terahertz frequencies. The protein structure could be a two-state structure or basically a multi-state structure. And this system model basically conveys a communication system model because it's composed of the three major elements: a transmitter, a channel, and a receiver. But what about the mechanism of operation? Well, the nano-antenna electromagnetic field, it results in the polarization of the protein molecules. Actually, in the system, if an external harmonic excitation has a frequency which matches one of the natural frequencies of the system, then resonance occurs. Therefore, the protein is going to be primed from a low energy state into a higher energy state. And as such, a chemical interaction is going to follow. So this electromagnetic protein interaction I'm talking about, it basically impacts the protein probability of being in a folded state. Now, a question that you might be wondering about is that how can I make sure that basically I'm targeting the protein of interest and not any other protein? How can I make sure of how selective I am and how controllable is basically my mechanism? For that, our major contribution is that we have developed a selectivity metric, because if I want to target a protein population, I'm expected to affect the conformational change in the desired receptor molecules in order to induce a particular functional response. This is why we wanted to develop a metric that is going to provide a single value to quantitatively decide whether or not our interaction is sufficiently controllable. A maximum selectivity score is going to be expected when only the targeted population experiences a folding behavior due to the impinging terahertz signal. This analysis lays the ground for directed signaling, where desired proteins are driven towards a conformation that evokes a particular response. But how can we mathematically formulate such a metric? Well, we need to think of two things: how to model the protein structure, as well as how to model the protein dynamics. So we first modeled the protein structure as a harmonic oscillator, and we modeled the protein dynamics using the Langevin equation, which is a second-order differential equation. By solving the Langevin equation, we were capable of deriving the steady-state energy of the driven protein motion. And then we used Boltzmann distribution because it provides the probability that a system is in a particular state as a function of the system's energy. The protein probability of being in a folded state is mathematically given by the following expression, where we can see that basically it relies on the induced energy that have triggered it. Well, then from the probability of being in a folded state, we can come up with our selectivity metric, which is basically given as follows. In the numerator, we have the difference between the probability of the folded state of the desired protein and the probability of the folded state of the undesired protein. And in the denominator, we have a maximization function, which serves normalization purposes. And since our selectivity metric basically depends on this difference, which relies on the probability of being in a folded state, it means that it captures both the system mechanical parameters as well as the amount of energy that is basically stored in the terahertz stimulated protein. So let me show you what our results look like. First of all, we consider two protein molecules in the vicinity of one another to mimic an intra-body environment. Those proteins are rhodopsin and bacteriorhodopsin. We consider a population size of 1,000, and we have the following protein properties from the frequency, stiffness, as well as mass. So we have here figure 1, which presents basically a pseudo color plot. In my x-axis, I have the force. In my y-axis, I basically have the frequency, and the color map is for the selectivity score. We have two populations, rhodopsin and bacteriorhodopsin, where rhodopsin is basically the targeted population. This is why we can see rhodopsin, the selectivity score is above 0, while for bacteriorhodopsin, it is basically below 0, signifying that it is basically the untargeted protein population. Interestingly, we can see that there is a specific point at which my system achieves maximum selectivity score. This occurs at a specific frequency as well as at a specific force. This frequency is basically the vibrational frequency of rhodopsin, which occurs at 1.36 terahertz. The same thing occurs if we generalized our model and we considered 3 protein populations, where I'm targeting basically population 2 as seen in figure 2. I have also on my x-axis my force, on my y-axis the frequency, and my color map is basically the selectivity score. And also I have at a certain force and a frequency, I can get basically a maximum selectivity in the system. So basically this tells my experimenters that if you can develop basically a nano antenna tuned to that vibrational frequency, and we can have the following force, then we can guarantee a maximum selectivity in the system, which means that we can control our interactions. The pseudo-color plot is also a powerful tool because it shows what regions should be targeted and those that should be avoided in order not to activate the wrong protein population in the system. So basically, this brings me towards the end of my presentation, where I'm going to leave you with 3 key messages. An important characteristic associated with the protein structure is that it exhibits vibrational spectral features in the terahertz regime, corresponding to functionally relevant modes. The developed selectivity metric measures the capability of the nanoantenna to stimulate the desired protein population to change its conformation without impacting the conformation of other proteins in the system. The selectivity metric provides a score between -1 and 1, where the closer the value achieved to 1, the higher the selectivity in the system. Thank you very much, and I'll be very happy to answer any of your questions.
Great, thank you very much, Hadeel. So I'll ask a question and give people time to type theirs. Hadeel, what happens to the selectivity metric if a higher force is used on the system?
Okay, that's actually a very good question. What happens is that if we pump more force in the system, we are going to activate the untargeted protein population. Actually, I have a visualization for that. So if I put a lot of force in the system, both the targeted and untargeted protein population are going to be into a folded state, and hence the selectivity value is going to drop. This is why there should be actually an optimal force value that is going to guarantee that only the targeted protein population goes into the folded state, and as such a maximum selectivity score is achieved.
Great, thank you. And what's the range in terahertz that the protein vibration frequency generally spans?
So based on the literature, the protein vibrational frequency, it ranges between 0.03 terahertz to 6 terahertz, basically.
Great, thank you. Marshall, are there any questions?
Yes, we have a written question, which I'll read directly. And this person says, thank you for your talk. Have you tried your system in biological samples yet where multiple proteins are present?
So thank you actually for this question. I would love to, but I have been working on the theoretical model itself. So I'm working on the part of the theory where we are developing basically those metrics, but I would love to take, like this work and move it into some sort of experimentations if the time is permitted, like during my PhD. So yeah.